Good morning students. Welcome to Leela's tutorial. So in last lecture we have discussed about stem cells and their properties. And coming to the types of stem cells, we said it is two types, that is embryonic stem cells and the adult stem cells. So let us see what are the embryonic stem cells and how they are cultured in vitro. So here it is the culturing of embryonic stem cells. The embryonic stem cells we are saying. That means they are obtained from the developing embryo, right? So from the developing embryo, from where we are taking or from where you are taking the cells or the harvesting the cells. So here you can see these cells are derived or obtained from the inner cell mass of blastocyst. So here this is one of the developing stage of the embryo where this is the blastocyst. So the inner mass of cells you will be collecting and use as a stem cell. So here these are referred as embryonic stem cells as they are obtained from the one of the stage, that is the blastocyst stage of developing embryo, right? So these stem cells are used or can be cultured in order to obtain a complete baby or the complete organism or a specific tissue or organ, right? So here let us see how you are going to culture these embryonic stem cells. So let us see from the development of the embryo. Here you can see the male gamete, that is the sperm cell, and here you have the female gamete, which is the egg cell. So here the haploid cells, right, the haploid cells, when they are getting fertilized, when the egg cell is getting fertilized with the sperm cell, it transforms into a diploid zygote, right? So here after fertilization you can see it has developed into a zygote. This will be the day 1. The first day after fertilization it will form a zygote, where here you can see it is having an totipotency. So, or you can say the zygote is an totipotent cell. What do you mean by totipotent? Totipotent is nothing but it's the ability of the single cell to get transformed into a complete organism, right? So now you can say that this single celled zygote at the day 1 stage is a totipotent cell which is having an ability to transform into a complete baby, right? So here after first day, the second day it will be the two-celled embryo. On 2 cells will be formed due to cell division. Then next it will be at the third day, it is 4th cell stage where the embryo consists of 4 cells. Now as the days pass, right, the cell division increases. That means the cell is undergoing the differentiation process as the days increase. So here you can see at day 4 it is called as morula stage, where it consists of 8 cells, right? Then after that, with several divisions, with several cell divisions, the next cell or the next stage is referred as blastocyst stage, where the blastocyst consists of 2 types of cells. The outer lining of the blastocyst is referred as blastomeres or the blastomere cells, right? And here you can see a mass of cells. So these mass of cells are still undifferentiated stem cells. So these undifferentiated stem cells are used as these undifferentiated stem cells are used as your embryonic stem cells. Is that clear? So now if you want an embryonic stem cell, what you will do? From where you will be deriving or from where you will be extracting them? You will be extracting from the developing blastocyst stage. From this blastocyst stage, you are going to collect or derive the mass of cells. Then you are going for the further culture process. So here you are going to harvest the cells in vitro. What do you mean by harvesting? Harvesting is nothing but culturing the cells. You are providing the nutrition in terms of animal tissue culture media and you are growing the cells in vitro. In vitro is nothing but outside the body, right? Here the baby or the complete cell differentiation is not occurring inside the body, right? You are extracting the cells outside in the lab. So in vitro is referred as working in the lab or outside the body. So you are harvesting in vitro and culturing in suitable animal tissue culture media or animal cell culture media, right? So here you have the cell culture media and you have the harvested cells. You have harvested and culturing them in vitro, right? You are not culturing inside the body. You are culturing them inside the container or the animal tissue culture vessel, right? So here all the environmental conditions are maintained, the suitable temperature, pH, the media, the nutrients required, everything will be provided for your cells in order to undergo the differentiation process. So this is the culture process, right? You are going to harvest the mass of cells from the blastocyst stage, right? And then you are going to grow them in a suitable animal tissue culture media under controlled environmental conditions. So here the cells as they are obtained from the developing stage, right? They require some ready-made nutrition, right? The sufficient nutrition must be supplied. So here you can see a lining of cells are present which is adhered to the substrate. So here, or you can say that the cells are coated to the substrate. Here in animal biotechnology, substrate is the container, right? The lining of the vessel to which the cells go and adhere. When the cells adhere, when they are anchorage dependent cells, right? So here the container is coated with the feeder cells. Okay, mostly the feeder cells are fibroblast cells. So these feeder cells provide sufficient nutrients, right? They provide sufficient nutrients for the growth of the embryonic stem cells. Okay, so here the feeder cells are providing extra nutrition. In animal cell culture media also you have the nutrients, but still the ready-made nutrition is provided by the feeder cells, which are nothing but your fibroblast cells. So here the fibroblast cell lining is present in the container. Then you are going to add the media and the harvested cells are introduced into the media. Then after inoculation, right, you have inoculated the cells in the media. After inoculation, it will be the incubation, right? Incubation for several times. So now after this incubation process, you have to decide what type of cells you want to produce. By using the embryonic stem cells, what type of cell you want. If it is an skin cell or the nerve cell or bones, muscles, liver, thyroid or any other cell, based on that you are going to allow these cells to get differentiated into ectoderm, mesoderm or endoderm. Is that clear? So here once they get differentiated, now see here from this mass of cells or the blastocyst stage, till here you have till here you have only one type of cells, right? But here you have two types of cells where it is the blastomeal lining and also the mass of cells, right? That means some differentiation process has occurred. The stem cells are undifferentiated cells which doesn't have any structure or the function, right? As these stages increase, the embryo will undergo differentiation to form different types of lineages, right? Or different types of cells. Now you call them as pluripotent stem cells. So now these pluripotent stem cells will undergo further differentiation to form a multipotent or to develop into an multipotent cells which have ability to develop into any types of cells. Is that clear? So in that way you are going to develop whatever the cell required. If the person want to produce some skin cells, right, because of some skin burn accident or something, he wants the skin cells to be produced. So what he can do, he can go for the embryonic stem cultures technique and he can produce that. If it is having some bone defect, then again the bones. What type of cells, even for the research, if you want to work on any type of the cell, that specific type of cell can be produced by this embryonic stem cell culture technique. Is that clear? So here the main thing is the inner mass of blastocyst stage cells will act as your embryonic stem cells. You need to provide all the sufficient nutrients and the feeder layer to them, and then allow them to undergo differentiation process into pluripotent cells, then they develop into multipotent cells, and then you get the required type of cells which you want to produce. Is that clear? This is about your embryonic stem cells. In next lecture, we'll see production or culturing of adult stem cells. If you have any doubts, put it in comments. Okay. Subscribe for further lecture.